the evolution of most phenotypes are largely unknown. And over the last few years, I have been privileged to play a leadership role in the Xenomia Consortium, which has sequenced and aligned the genomes of 240 mammals. So the question I wanted to ask is, how can we use this new alignment of hundreds of mammalian genomes to investigate phenotypic diversity? And I'm going to focus for the duration of this talk on my specific project that I led within Xenomia, but you can read a summary of all of our projects in our paper that came out in Science last April. So for most of this talk, I'm going to focus on the phenotype of large brains relative to body size. And this phenotype has evolved multiple times. So humans have much larger brains than cats and rats, and even than our closest relatives, chimpanzees. Yet dolphins, which are much further from us than chimpanzees are, also have large brains. So we can plot body weight on the x-axis and brain weight on the y-axis, and we see that there's generally a linear relationship, but that humans have a much larger brain weight than we'd expect based on their body weight, as do dolphins. But the blue whale, which is much closer to dolphins than dolphins are to human, has a much smaller brain than we'd expect relative to its enormous body. And while my passion is understanding the mechanisms underlying phenotype evolution, studying these mechanisms has the potential to provide insights into human disorders related to the phenotype, such as microcephaly, which is being born with a brain that's too small. So for the duration of this talk, I'm going to refer to this deviation of the expected linear relationship of body weight versus brain weight as the brain size residual. So while I was working on Xenomia, a paper came out that curated brain size residual phenotype annotations for a huge number of mammals. And I want to point out that this data set contains over 150 of the mammals in Xenomia. And I also want to point out that the largest variation in brain size residual is in the cetaceans, which are the whales, and that this species with the largest brain size residual is the human. So then the question we asked is, how can we use these genome sequences to study the evolution of brain size residual? And the first thing that we thought to do was to compare protein coding sequences. So my lab mate Morgan Worthland did this, and unfortunately, she was not able to find any protein coding regions associated with the evolution of brain size residual. And what this suggests is that brain size residual has evolved at least in part through gene expression. So in this toy example, we have an enhancer bound by a transcription factor that loops in 3 dimensions to a transcription start site, recruiting RNA polymerase. And this enhancer is activating gene expression in the brain, enabling humans to have large brains. And now if we consider our closest relative, the chimpanzee, with a smaller brain size residual, there might be a difference in sequence in the enhancer that causes a difference in transcription factor binding, that then causes a decrease in transcription of the gene in the brain, which then leads chimpanzees to have smaller brain size residuals. This suggests that there are actually multiple approaches that we can take to using genome sequences to study phenotypic diversity. First, as I mentioned, we can compare protein coding sequences, but we can also potentially learn a lot from comparing enhancer sequences. So a previous study found a candidate enhancer associated with the evolution of brain size residual, and this is an enhancer in the chimpanzee whose ortholog in the human is lost. So now that we have a huge number of cetaceans or whales that have been sequenced with a huge variation in brain size residual, I asked myself, is this enhancer also lost in the cetaceans with large brain size residuals? And to my surprise, what I found is that this enhancer actually maps to all of the cetaceans in Xenomia, including those with large brain size residuals. And what this suggests is, if we want to fully understand how brain size residuals evolved, we have to study brain enhancer activity across a much more diverse set of species than was available in this earlier study back in 2011. So a previous method was developed to find enhancers associated with phenotype evolution. And this is a nucleotide-level conservation-based approach. It's called RAR converge, and it was previously used to study the evolution of blindness in subterranean mammals. So I then decided to check, can we use this method to find enhancers associated with the evolution of brain size residual? And doing this, I found exactly one associate enhancer. And when I looked in a human Hi-C or 3-dimensional DNA interaction data set, it did not interact with any transcription start sites of any genes known to be involved in brain size, suggesting this enhancer is also not the entire brain size story. However, I thought there still might be more enhancers involved in the evolution of brain size residual. And the reason I thought this is that nucleotide level sequence conservation is not always indicative of enhancer activity conservation. So there are a number of reasons for this. One of them is that most mammalian enhancers have tissue-specific activity. After all, their job is to determine the specific tissues in which genes are expressed. But conservation information is tissue agnostic. The other reason is that enhancer function can be conserved when nucleotide level conservation is low. A wonderful example of this was published a few years ago of a candidate enhancer in humans whose ortholog or corresponding region in zebrafish is an enhancer, and even its ortholog corresponding regions in sponges is an enhancer. Now, these different colored boxes are binding sites for different transcription factors. And what you see is that human, zebrafish, and sponges all have the same colored boxes, which allows the enhancer activity to be conserved, but the boxes are in different orders in different species. So if you map these nucleotide by nucleotide, you would not see especially high nucleotide level sequence conservation. What this does indicate, however, is that sequence feature conservation is indicative of tissue-specific enhancer activity conservation, and sequence features can be transcription factor binding sites. Thus, how should we be identifying enhancers associated with phenotypes like brain size that seem to evolve through gene expression? As I mentioned, while nucleotide level conservation has led to exciting results, it is often too limited for us to find all of the relevant enhancers. Another option is to experimentally assay enhancer activity in a tissue related to our phenotype. But the reality is that we cannot obtain most tissues from most mammals. And even if we could, that's not something we want to do. So that is not a great choice. As a postdoc, I developed a research program to efficiently trace the evolution of phenotypes that have evolved through gene expression. And what I mean by efficiently is without having to sacrifice large numbers of mammals. So the high-level idea, which may not be a surprise to this particular audience, is to use machine learning. So what we can often do is generate enhancer activity data from a relevant tissue in a small number of species. So we have a matrix like this, where each row is a candidate enhancer, each column is a species. Most of our data is missing, but we have data from a few species. What we can then do is take sequences underlying enhancers and non-enhancers and use them to train a machine learning model, and then make predictions at the orthologous or corresponding regions, the enhancers we identified, in a much larger number of species, allowing us to fill in most of this matrix. And we call this framework for doing this TACIT, and I'll explain why in a few slides. I'm going to begin by telling you about the TACIT methodology. I'll then describe evaluation criteria that we developed for evaluating machine learning models for TACIT. After that, I'm going to show you some of our results for applying TACIT to brain size. And finally, I'm going to tell you about some of my plans in the future to apply TACIT to other phenotypes. And I should mention as I go that you can feel free to ask me questions as they come up. You don't have to wait till the end, though you also are welcome to wait till the end. So let me begin with the TACIT methodology. So the first thing we need to do is identify candidate enhancers in a species relevant to our phenotype. After that, we want to take sequences underlying enhancers and non-enhancers and train a machine learning model to predict enhancer activity in our tissue. We then want to make predictions and enhancer orthologs or corresponding regions of the enhancers we identified across a large number of species. After that, we want to associate our predictions with the phenotype. And finally, we want to evaluate the statistical significance of our association. So we call this approach the Tissue-Aware Conservation Inference Toolkit, or TACIT. I'm now going to tell you in a little more detail about exactly how we applied TACIT to study the evolution of brain size residual, beginning with the first step of TACIT. So my lab mates, Morgan Worthlin, Alyssa Lawler, and Ashley Brown, generated a TACIT data from 4 different mammals of different levels of relatedness in the region of the brain called the motor cortex. And for those of you not familiar with the TACIT, the idea is that an enzyme comes in and chops up DNA, but most of your DNA is wrapped around nucleosomes, and it can only access the DNA that is not wrapped around nucleosomes or open. And these regions that are not wrapped around nucleosomes can more easily be bound by transcription factors, allowing them to be more likely to function as enhancers. So what we get is when we chop up the DNA from a large number of cells, we can then map it to the genome, and we get these piles of pieces of DNA, which we call ataxic peaks. So here we have one gene, and then here in this toy example, we have 2 more, which are candidate enhancers. And while these ataxic peaks. Not all of them are enhancers, and ataxic does miss some enhancers. For the purposes of this talk, I'm going to consider these ataxic peaks to be candidate enhancers. So now that we have our data, I'm going to tell you about how we trained our machine learning models. So the first decision that we decided to do was to use a machine learning model approach called convolutional neural networks, an approach I know is near and dear to this audience. Then we had to decide how we wanted to define our positive and negative sets. So for the positive sets, we took our brain enhancers, which were our ataxic peaks, and specifically, we took the peak summits plus or minus 250 base pairs, but that still left the question of what negative set should we be using. And what I'll tell you is, from my experience, choosing a negative set that is relevant to the task at hand is actually more important for getting a model to work well than the exact architecture of the CNN. So what I decided to do first was to try out a number of negative sets that have been published in previous papers, including some papers by, I think, people on this call. And I don't have time to go describe all of the results for all of these in this talk. You can ask me about them offline, and I'm going to focus on one specific negative set for this talk, and that is the nucleotide shuffled enhancers. These are enhancer regions where what we did is we shuffled them in a way that preserves the frequency of pairs of nucleotides, and we chose 10 negatives for every one positive. However, I then realized that these previous approaches were not designed specifically for the task at hand, because what our goal is to do is to take an ortholog or corresponding region of enhancer and predict whether that ortholog is an enhancer. Thus, I developed a new negative set directly for this task, and I call these non-enhancer orthologs of enhancers. These are regions in closed chromatin whose orthologs in another species for which we have data are open. So then the question was, how do I get these orthologs of enhancers? So to do this, I used a publicly available method called HAL liftover, designed to work on the alignments made by Zoonomia, with a post-processing tool that I developed with an amazing former undergraduate of mine named Irene, and we call this tool Helper. You can check this tool out in our GitHub. You can read about it in our paper in bioinformatics. And I'm really excited that even though Irene had never done research before working with me, she's decided to continue pursuing a research career and is currently a PhD student at the University of Illinois at Urbana-Champaign. So now that I've told you about how I created my negative sets, I'm going to describe first our results, and I'm going to focus on results trained for machine learning models trained using only DNA sequences from the house mouse. And the reason that I started with only training models with data from one species is if I want to know I can trust my machine learning models in a species not used in training, I first need to train my models using a strict subset of the species for which I have data, and then evaluate the performance in species of different levels of genetic relatedness. So first, here what you can see is the performance for all of our negative sets on chromosomes of the mouse genome not used in training. I have the AUC and the AUPRC, and what I want to point out is a few things. First, all the models trained on all the negative sets on parts of the mouse genome not used in training work really well. And second, the model that gives the best performance by a small amount is the one trained on the dinucleotide shuffled enhancer negative set. However, as I mentioned, our goal is not to evaluate, do you have a model that works amazingly on chromosomes not used in training? Our goal is to actually make accurate predictions in a new species that the model hasn't seen. So to do this, I evaluated my models trained on house mouse on all the other species for which I have data. For the purposes of this talk, I'm going to tell you about our results for evaluating in rhesus macaque, which is a type of monkey, and I'd be happy to tell you about our results for evaluating other species offline. So to do this, I'm going to take a short deviation from the TACIT methodology and tell you about how we evaluate machine learning models for TACIT. So the next few slides, I'm going to be illustrating results as violin plots. You can think of violin plots as smooth histograms. And something specifically that I want to point out is that all of our predictions are in the 0 to 1 range, which is the range indicated by this dark colored line in the middle of the violin plot. But because of the smoothing, sometimes they appear to be out of that range, even though they are not actually out of that range. So the first set that I evaluated on is what I call macaque plus mouse. These are candidate enhancers or open chromatin regions in the macaque whose orthologs in the mouse are also open. And a model doing well on this would mean that the model can accurately predict that enhancer activity in the brain is conserved. And because we expect a positive, I've indicated that as a plus at the top of the violin plot. So first, what I want to point out is that FAST-Con scores, is the metric of conservation scores that varies from 0 to 1, tended to be low for these regions, even though the enhancer activity is conserved, thus demonstrating in a systematic way that nucleotide level conservation often fails to pick up enhancer activity conservation. I should also mention that I did these evaluations also for phylop scores. I don't have time to go into the results in this talk. That's another method of nucleotide level conservation, and the results tended to be slightly worse than they were for FAST-Con scores. In contrast, our machine learning models most of the time accurately predict that these macaque enhancers are candidate enhancers, showing that our machine learning models can identify enhancer activity conservation even when nucleotide level conservation is low. However, as I mentioned, our long-term goal is to understand phenotypic differences between species. And to do that, we don't just need to evaluate on enhancers whose activity is conserved, we also need to evaluate on enhancers whose activity differs between species, because it is these enhancers that are likely to play a role in phenotype evolution. And I should mention here that Stein-Ertz's lab had a paper a couple of years back showing a beautiful example of an enhancer whose activity differs between species that their models were accurately able to predict, which was an important foundation for our work. So I wanted to develop a way to evaluate this systematically in a genome-wide way. So to do this, I developed two new evaluation criteria. One is what I call species-specific enhancers and non-enhancers. These are candidate enhancers whose orthologs in another species for which we have data that is closely related are not enhancers. The second is what I call clade-specific enhancers and non-enhancers. And these are candidate enhancers in a clade for which we have data, whose orthologs in another clade for which we do not have data are not enhancers. Since time is limited, I'm going to focus this talk on our results for the clade-specific enhancers and non-enhancers, and I'm happy to describe our results for species-specific enhancers and non-enhancers offline. And to do this, I took models trained in the house mouse and evaluated them in the rhesus macaque. So the next evaluation I'm going to show is what I call macaque not equal to mouse. These are candidate enhancers in the macaque whose orthologs in mouse are not enhancers. And our models doing well on those show that they can predict that a region is enhancer even when the ortholog or corresponding region in another species is not an enhancer. And once again, the plus on the top of the violin plot indicates that since we're evaluating on the macaque sequence, which is an enhancer, we expect a positive prediction. And I was really pleased to see that the models trained across multiple negative sets do really well on this task. The next evaluation criteria I'm going to describe is what I call mouse not equal to macaque. These are closed chromatin regions or non-enhancers in the macaque whose orthologs in mouse are enhancers. So this is showing whether we can train a model on one species, and if an enhancer is lost in the other, can we accurately predict that loss in enhancer of activity? And what I've indicated by a minus on the top of the violin plot is here, since we're evaluating on the macaque species, which are non-enhancers, we'd expect a negative prediction. Very disappointingly, the model trained on the dinucleotide shuffled negative sequences, which did so well on regions in the mouse genome not used in training, almost half the time incorrectly predicted that these non-enhancers in macaque were enhancers. In contrast, the model trained on the negative set that I developed specifically for this task most of the time correctly predicted that these non-enhancers in macaque are not enhancers. And I should notice here that the FAST-Con scores tend to be low, which means that there's low nucleotide level sequence conservation, but they do not actually tell us whether the region is actually functional in some species, and if so, in what species it has a function. In addition to being able to accurately predict enhancer activity differences between species, we wanted to see if our models had overcome conservation scores limitation of tissue agnosticity. So in other words, we wanted to see if we have an enhancer in a tissue that's not brain, such as liver, enhancer in brain that is not enhancer in non-brain tissue, such as liver, we wanted to see if we could accurately predict that this region is not an enhancer in the liver. So to do this, once again, we took models trained in house mouse and evaluated them in rhesus macaque, and we also took models trained in the brain and evaluated them in the liver. So the next evaluation I'm going to show you is what I call brain non-equal to liver. These are candidate enhancers in the brain that are not enhancers in the liver. And our model doing well on these, since our model's goal is to predict brain enhancer activity, shows that our models can predict brain-specific enhancers relative to another tissue. We were really pleased to see that models trained on multiple negative sets did really well on this task. The next evaluation I did is what I call brain plus liver. These are candidate enhancers in the brain that are also enhancers in the liver. And our model doing well on these shows that that our models can not only learn in sequence signatures of brain-specific enhancer activity, but can also learn sequence signatures of enhancer activity that is shared between two very different tissues. I was really pleased to see that models I trained on multiple negative sets do really well on this task. Finally, I evaluated what I call liver not equal to brain. These are candidate enhancers in the liver that are not enhancers in the brain. And thus, since they are not enhancers in the brain and the model's goal is to predict brain enhancers, I would expect the model should be predicting a negative, is indicated by a minus at the top of the violin plot. Very disappointingly, the models I trained on the dinucleotide shuffled negatives almost half the time incorrectly predicted that these liver enhancers that were not brain enhancers were actually brain enhancers. In contrast, the model I trained to predict directly enhancer activity differences between species with the new negative set I developed for this task usually correctly predicted that these non-enhancers in the liver, enhancers in the liver that are not brain enhancers are not brain enhancers. And I think what's going on here is that even though I wasn't explicitly putting liver enhancers that weren't brain enhancers into the negative set, many times if you have an enhancer that's conserved enough that you can map it across species, but not conserved enough that its activity is conserved, might actually have activity in, say, multiple species where you measured it in your tissue of interest, and then in multiple tissues, where you measured it, and then in the orthologous species, maybe only a strict subset of those tissues. So thus, my model setup was able to learn this tissue specificity of enhancer activity. I should also point out that the FASTCON scores, because they're tissue agnostic, are not especially useful in telling us any information about tissue specificity of enhancer activity. So you can learn more about these evaluation criteria, as well as additional evaluation criteria we developed and performance on additional models I trained using additional negative sets in my paper that came out last year in BMC Genomics. So here we have finally the performance of our final models we used, where we took all of the motor cortex enhancer activity data, put it together, and trained one large model. And this is our test set performance. The white bars for the AUPRC indicate what we'd expect at random, and you can see that our final model works extremely well. So now that I've told you about how we evaluate machine learning models for TACIT, I'm going to return to telling you about the rest of the TACIT methodology. And to do this, I next am going to explain how we made predictions across large numbers of species. So the way that we did this is by using the Xenomia alignment of hundreds of mammals developed using the cactus algorithm. And specifically, we decided to focus on the Boreoeutherians, which are the mammals indicated in the black boxes. And our reason for limiting ourselves to the Boreoeutherians is that all of the ATAC-seq data we have comes from Boreoeutherians. So we cannot actually evaluate the performance of our model in more distantly related species. So now that we have our candidate enhancers in the motor cortex, predictions in 222 Boreoeutherians from Xenomia, we needed to find a way to associate these predictions with a phenotype. So to describe how we did this, I'm going to go into the fourth step of TACIT. To motivate our approach for doing this, I want us to consider this toy example of a macaque enhancer. And you can see here its orthologs and a number of other mammals and the predictions of those orthologs, as well as our phenotype annotation. And what you notice at first glance is that there's an extremely strong positive association between our predictions and our phenotypes, suggesting this candidate enhancer should be a high priority for follow-up studies of brain size residual evolution. However, if we look a little more carefully, what we can see is there's also a very strong negative association between the predicted enhancer activity and the distance between species and the rhesus macaque. So it is impossible to know if this enhancer is actually involved in our phenotype, or if this entire result is due to the confounding factor of the phylogeny. We therefore decided to use a method called phylogeny to evaluate associations between enhancer activity and our phenotype. And phylogeny is a sped up version of phylogenetic generalized least squares. So the idea of this is we have our candidate enhancers. We use a generalized least squares regression to evaluate the association with our phenotype, but we have an expected variance covariance matrix that's based on the phylogenetic relationships between species, so that associations we find cannot be entirely explained by the phylogenetic tree. And for binary phenotypes, we use the logistic version of phylum called phyloGLM. So now that I've told you about how we evaluate our associations, I'm going to describe how we compute our statistical significance. So to do this, we used a recently published method called phylogenetic permutations. And the way this works is we want to create a null distribution by permuting our phenotypes many times. We want to do it in a way that preserves the higher order level of the phylogenetic tree so that we have a more realistic null. And fortunately, as we realized we need to do this, our colleagues at the University of Pittsburgh put out this method that does exactly this, called phylogenetic permutations. So I asked a fantastic former undergraduate in the Fenning lab, Daniel Schaefer, to run this for about 100,000 motor cortex enhancers for a few different neurological phenotypes. And he came back to me and he said, this method is way too slow, and it's especially too slow for binary phenotypes. He then implemented a speed up that substantially decreased the runtime. And for binary phenotypes, the speed up was over an order of magnitude. You can check out the speed up on our GitHub. And I'm really pleased that Daniel is also continuing his career in computational biology research as a PhD student at MIT. So now that I've told you about the TACIT methodology, I'm going to tell you about some of our results from applying TACIT to brain size. So using TACIT, we identified 49 motor cortex candidate enhancers associated with the evolution of brain size residual. So Daniel and I worked together to do a massive literature review on the genes near these enhancers. And we found that 14 are near genes previously implicated in micro or macrocephaly, so having a brain that's too large or too small. So I then asked the question, do candidate enhancers near genes previously implicated in micro macrocephaly tend to have more significant associations with the evolution of brain size residual than other candidate motor cortex enhancers? And the answer is that they do. And there's a significant trend in that direction. And I also did this using the RER converge results. That's the method, the nucleotide-level conservation-based approach, and did not find this trend. So here you can see an example of a candidate enhancer associated with brain size residual. On the x-axis, we have our predicted motor cortex enhancer activity. On the y-axis, we have the brain size residual. For the purpose of not crowding this plot, I have just plotted the results for the cetaceans, which have the largest variation in brain size residual, but the mechanisms underlying brain size residual evolution in cetaceans were previously unstudied. And we can see here is a really strong negative association. So here we can look a little more closely at our cetaceans. We have our predictions and we have our brain size residual annotations. So we can look at the bottlenose dolphin, which has a large brain size residual, and its closest relative, the killer in zoonomia alignment, the killer whale with a smaller brain size residual. And we see a large difference in our enhancer activity prediction in the opposite direction. We can then look at these two other whales with large differences in brain size residual that are closely related to each other. And once again, we see a large difference in predicted brain enhancer activity in the opposite direction. Finally, we can look at our two different species of minke whales. Both have large negative brain size residuals, so much smaller brains than we'd expect relative to their body size, but one is more negative than the other. And once again, we see a difference in predicted enhancer activity in the opposite direction. So we then asked, where is this enhancer? So here we have a browser shot from the common minke whale. These black hash marks are orthologs of candidate enhancers, and the one in the red box is the one that's associated with brain size residual. And you can see that these are near the gene LRIG1. So then I asked the question, is this candidate enhancer actually interacting with the transcription start site of LRIG1? So to do this, Daniel looked in a three-dimensional DNA interaction data set from the adult mouse cortex, and we chose to focus on mice because they have small brains, and this enhancer is negatively associated with brain size residual. And what we found is that there is a statistically significant interaction with LRIG1's transcription start site. And we were really excited about this result because this data set was actually really shallow, so we weren't sure if we'd find any interactions of this enhancer with anything, and we were really excited that we actually found one with LRIG1. LRIG1 is expressed in the mouse cerebral cortex, and LRIG1 has been shown to regulate quiescence in adult neural stem cells and neural stem cell proliferation during development. So now that I've told you about some of our results from applying TACIT to brain size, I'm going to tell you about some work that I plan to do in the future in applying TACIT to other phenotypes. So one set of phenotypes that I'm really interested in is the evolution of immune phenotypes, including longevity relative to body size and infectious disease susceptibility. This is work that I'm currently working on remotely with an undergraduate in Andreas Fenning's lab. And to do this, we are using lymphoblastoid cell lines, which are derived from B cells, some data that's publicly available from multiple species. Another direction I'm really excited about pursuing is studying the evolution of metabolic phenotypes, such as dietary phenotypes like carnivory and herbivory and hibernation phenotypes. And to do this, I plan to focus on adipose and liver tissue. And finally, I'm interested in developing the computational methodology behind TACIT further. One specific area I'm interested in is looking at closely related species and individuals within a species. And I think that this is going to be especially challenging because a lot of the time we don't actually see that enhancer is gained or lost, but rather when we have ATAC-seq data from either a bulk tissue or a pool of cells from a single cell experiment, we actually have signal as a quantitative measure. And instead of seeing a gain or loss, we see a stronger enhancer becoming a weaker enhancer. So to do this, we're going to need to train regression models, which is something that many others have shown we can do in general to predict enhancer activity on regions of the same species not used in training, but we want to actually show that we can make accurate predictions of differences in enhancer activity between species, and this is going to require new evaluation criteria. In addition, I want to be able to apply this to more distantly related species than we had in the Boreoeutherians in Zoonomia. And one of the limiting assumptions currently of this method is that I assume that I have the same candidate enhancer involved in phenotype evolution in all of the species. And when we have more distantly related species, we're likely to have maybe different enhancers in different parts of the tree regulating either the same gene or genes in the same biological process. So I'm going to need new methods to account for that possibility. And this is something I'm really excited about because it's going to allow us to study those more distantly related species in Zoonomia, some of which have really cool hibernation patterns. Also, it's going to allow us to go into studying evolution of enhancer activity and phenotypes it controls in birds. There's now a large alignment of about 600 mammals and birds that I'm excited to use, as well as even more distantly related vertebrates, and I'm lucky to be involved in the Vertebrate Genomes Project for these investigations. And if any of this future work interests you, I would highly recommend reaching out to me about joining the lab. I'll be starting next fall at Carnegie Mellon. I'm recruiting PhD students from both computational biology and biological sciences, as well as postdocs and staff scientists, so please reach out. So in conclusion, we have developed TACIT, which associates predicted enhancer activity of candidate enhancer orthologs with phenotypes. Using TACIT, we identified 49 motor cortex candidate enhancers associated with the evolution of brain size residual. And as I didn't have time to mention this in my talk, but I'm happy to answer questions offline, we also found additional enhancers associated with the evolution of other neurological phenotypes. And TACIT can be applied to any phenotype with annotations in dozens of species and enhancer activity data in a relevant tissue or cell type from at least a few species. And if you're interested in learning more about TACIT, you can ask me questions after the talk or check out our paper that recently was published in Science. And I should also mention that TACIT is one of multiple projects I've been involved in applying machine learning to biology. Here you can see a list of the others. You're welcome to ask me questions about this offline. And I also have some ideas for extending some of these others that I plan to bring to my own lab. So with that, I'd like to thank my postdoc advisor, Andreas Fenning, the Fenning Lab members involved in this project, especially Daniel Schaefer, Alyssa Lawler, Morgan Worthland, and Ashley Brown. Our funding and compute resources are amazing collaborators, and I'd be happy to take any questions.
Thank you. So, yeah, questions? Do you see any on the yeah, I can start with one, which is maybe clarifying something. This picture is great. So, in TACIT, do you rely on alignments between candidate and answers?
So for training
we
the
just have to
model,
align in the first place, correct?
so for training the model, we don't directly use the alignment, but then for making predictions across large number of mammals, we do need an alignment. So ideally, a multi-species alignment. The cactus algorithm is state-of-the-art. It can account for things like inversions. Something that we've also done, if we have a species that's not in a multi-species alignment but has a pairwise alignment to something closely related that's in it, is to map through the multi-species alignment and then map through the pairwise alignment to that species, or vice versa. Does that make sense?
Yeah. So the training is result, and then later on you need it. Okay. And so in your future work, where you want to go even all the way to the pathway, would you also need then to predict them answers that don't necessarily align? Or
yeah, so that's one of the challenges, is often when you get out like past mammals into birds or even further, a lot of the enhancers, you just can't map them. So one thing I've thought about doing is potentially doing separate enhancer mappings in different species, looking at associations, and then somehow jointly modeling different enhancers that might be regulating the same gene or genes in the same biological process.
I have a question for the, you said that you often see that enhancers, they don't like stop being an enhancer, but they just become less accessible or more accessible.
Right.
So did you mean that, could you explain, or what do you think is the mechanism there? Does the motive change slightly, or is it?
This is especially common in closely related species where you see enhancers where you might have like one of your strongest open chromatin peaks, then you map it and it's a mid-level peak. So I think what's going on is that an enhancer maybe becomes accessible in a smaller subset of the cells within your tissue or cell type. And that might mean that maybe your general gene is expressed in, fewer cells. So maybe the gene's like less important in the other species, but still doing something. Something that I have also seen is an enhancer that's, say, enhancer in 2 different tissues in one species, and the ortholog is an enhancer in, say, only one of those tissues in another. So part of that enhancer is conserved because it needs to, continue to be enhancer in one tissue, but part of it's not conserved because it's not an enhancer in the other one anymore.
I have a question.
Mm-hmm.
So did you see what kind of correlations did you see when you're looking at, I guess, enhancer conservation across species and like the importance of the gene that enhancer regulates?
That's a great question. So one of the challenges is that we only had 3D genome interaction data from human and house mouse. So there were some that, we found in Egyptian fruit bat that were conserved in large numbers of species, but not in primates and rodents, and we couldn't so we can't be sure what it regulates. Also, our data sets for the 3D interaction data were sparse. So often, we might not see an interaction with the gene, but still could be regulating. So I should just say a caveat, it's a little hard to figure out. I did look at the enhancers, just the specific set that were predicted to be conserved across almost all of the mammals. And Daniel Schaefer did a huge amount of different gene ontology analyses, looking at the human orthologs to see if they overlap GWAS hits, so human genetic variants associated with diseases. He didn't really find any consistent pattern. For ones associated with brain size, as I mentioned, we found that they tended to occur near genes previously implicated in micro or macrocephaly, suggesting these are important genes in the actual development of brain size. I didn't have time to go into this. We also did an evaluation for vocal learning, and we found that some of the enhancers are near genes with differential expression between a handful of vocal learners and non-learners from previous studies, so showing importance in vocal learning. But there, in terms of like level of enhancer activity conservation versus nearby gene role, there was no clear pattern.
I think this research is really exciting, wonderful area. I hope we make lots of great discoveries. So good luck as you forge it forward. I'm wondering if there is an analog here for coding. I mean, you said you couldn't find coding associations with brain size. I kind of in my mind rephrased that to, we don't have the statistical methods to detect those, thinking that they still might exist. First, I wonder if you agree with that. Second, I wonder if you think some of the methods around AlphaFold, things like predicting properties of proteins or protein interactions, whether you think that putting phenotypes like that onto each species with machine learning might help reveal some additional interesting associations on the phylogenetic tree.
Thank you, David, for your question. Also for a lot of the foundational work you've done in this area that my work extended. So I think it's a great question about protein coding regions. So the method we used was also RER converged for the protein coding regions, which is also a nucleotide level conservation based approach. And a lot of the time, this does make sense for proteins because they are super highly conserved at the nucleotide by nucleotide level across mammals. But there are a number of cases where even at the protein level, we can have functional conservation and not sequence conservation, such as in like transcriptional activator domains within transcription factors and things like that. So I do think there is the potential for machine learning methods to also potentially predict conservation of protein function in cases where nucleotide level conservation is low. Something like AlphaFold is definitely an option. I haven't seen the existing methods actually evaluate whether they can predict protein coding function similarities between species or differences between species. It's possible I missed it because it's a little more outside my area. So that's an evaluation that if it's not done, would need to be done. But I do think that could be cool and definitely would be excited about using those to then find new proteins associated with phenotype evolution that weren't found by the traditional nucleotide approaches. So I think in some cases, we do actually have the methodology for finding protein coding sequences, but in some cases, you're right, machine learning could be really helpful.